Okay, everyone, so in the last chapter we looked at physical control of microorganisms in the environment, and now we're going to take a look more at chemical control of microorganisms in the environment. Okay, so this is also chapter 9 in the book, like physical control was, but I kind of separated that out for time's sake. So chemical control, and again, this is in the environment now, not in the body. We'll get to that in the next chapter. Okay, so chemical methods of microbial control. Well, first we have phenols and phenolics. Now, phenols have this OH group here, and then this bent ring that's part of the compound, part of the chemical. Now, phenols and phenolics, phenols will disrupt proteins and cell membranes. They were used historically during surgery and are still used extensively today. Examples: natural oils, pine oil, clove oil, pine sol, things like that. Multi-surface here, disinfectant. Lysol, okay? Lysol is two of those phenol, benzene rings attached to each other. Bisphenol here. So Lysol. And until recently, another one was triclosan, which this was the chemical formula of that. Now, triclosan was used extensively in some of these products here, and you might notice the orange color on some of those. That orange color came at least in part from that triclosan. Now, again, this was a phenol that was used for microbial control, but effective back in 2017, the FDA banned the use of 19 antiseptics in over-the-counter hand washes due to, quote, the impact on the development of bacterial resistance, among other things. And here was a quote from the director of the team that instituted this ban. Consumers may think antibacterial washes are more effective at preventing the spread of germs, but we have no scientific evidence that they are any better than plain soap and water. In fact, some data suggests that antibacterial ingredients may do more harm than good over the long term. So yes, everyone, there is no need to get antibacterial or antimicrobial soaps. Just use soap. Okay, it's going to be just as effective, and it's not going to lead to resistance. Okay, it's just as effective. Here were the things that were banned, and you'll notice here is triclosan. So with the banning of that, it's like goodbye orange hand soap. You're not really going to see that much anymore. Okay, so that's phenols. Like think Lysol, that's the big one, or pine sol. All of those are referring to phenols, okay? Disrupting proteins and cell membranes. Now, we also have alcohol. This can be ethyl alcohol, like the alcohol you'd cook or drink, or isopropanol, isopropyl alcohol. This is rubbing alcohol. Generally, you want a 70 to 95% solution, not 100% alcohol. Stronger is not always better, as we see here. This is looking at this bacteria here, Streptococcus pyogenes, and if we have 100% alcohol, G means growth. That bacteria was growing just fine in its presence after 10 all the way up to 50 seconds. There's growth. No growth at 95 and so on here. It gets too dilute, then there's growth again. You don't want 100% because for this to be effective, there has to be some water in there so it can get into the cell. Alcohols denature proteins. They dissolve the lipids in cell membranes and dry up, desiccate. They're also bactericidal, fungicidal, and virucidal. And remember, cytal, that's destruction, killing the bacteria, fungi, or virus. And used extensively in degerming, as we see here. Remember, degerming, mechanical removal of microbes from a limited area, as we're seeing here. Alcohol, however, everyone, is not effective against endospores or fungal spores, so it cannot be used to achieve sterilization. Now, halogens. What are halogens? These elements in red on the periodic table. I mean, this last one is an halogen, astatine, but it's very, very rare and not used. But these other four are fluorine or fluoride, chlorine, bromine, and iodine. Okay? Halogens denature proteins. They are bactericidal and fungicidal. Interestingly, Disney World uses bromine to disinfect the water in their rides. So if you've ever been there, the water smells different than what you might be used to, say, in a pool that's chlorinated. That chlorine smell in like somebody's pool in their backyard has a different smell than the bromine at Disney World. Halogens can kill some endospores, some protozoan cysts, and many viruses, so they're fairly effective here. Of course, we've heard about fluoride in water and fluoride in toothpaste, things like that. How are they used? Iodine, which you might have heard as iodophores, or a tincture of iodine. You might have heard of betadine, especially if you're going into nursing. Betadine, pictured here, is a scrub, okay? It's quite effective. It continues killing microorganisms while in contact with the skin. Chlorine can be a solid, liquid, or gas, often put into water. Water supplies. Chlorine is highly effective as an antimicrobial. I mean, almost universally known is Clorox, chlorine bleach, hypochlorous acid, chlorine bleach. 2 drops in a liter of water is often sufficient. So picture a liter, you got a 2-liter bottle of soda, half that. You only need 2 drops in there is often enough, and 10% bleach can sterilize. It can kill endospores. So it's highly effective. It can be used in a gaseous form also to sterilize. Hydrogen peroxide is another one here. So moving on from halogens. Hydrogen peroxide releases oxygen radicals to damage cells. Remember this plate we saw in a previous chapter. You put hydrogen peroxide on that bacteria, the enzyme catalase or peroxidase is working, and we release that oxygen. These oxygen radicals can damage cells. How is it used? It can be used as an antiseptic, but interestingly, hydrogen peroxide actually kills more reliably on inanimate objects than wounds. It'll work on superficial wounds, but not so much deeper ones. It works great on inanimate objects, though. It can also be used as a disinfectant, and under the right conditions can kill endospores. Detergents. So you see this detergent here. Powers through tough grease and grime. Kills 99.9% of germs in 60 seconds. Now notice that in the ingredients list of this detergent, 98.8, so almost 99% of it's inert ingredients. So those are ingredients that are not really doing anything. They're just to give it substance. The active ingredient is this thing here, N-alkyl dimethyl benzyl ammonium chloride, and it's about 1.2%. Now that's a huge mouthful. It's basically this ammonium ion along with benzalkonium chloride here. That's the active ingredient in detergents that does this 99.9% of germs in 60 seconds. Detergents act as a surfactant because they are soap-based and an antimicrobial, probably the most common antimicrobial we use in our homes. Think about various detergents. Now, not surprisingly, some microbes like Pseudomonas are resistant to detergent. So we have to be careful there. Heavy metals is our next category, particularly Ag, that's silver, Cu, copper, Hg is mercury, okay? Heavy metals can often have antimicrobial properties, as shown here. We see the bacteria on this agar plate. These two aren't really destroying much, but this silver here is antimicrobial, okay? So we see that antimicrobial property there. Heavy metals denature proteins. Usually don't kill outright, though. They're bacteriostatic or fungistatic. How are they used? Silver and silver nitrate are particularly used a lot clinically and in the medical industry. Silver has antiseptic and disinfectant properties, and it is used to treat wounds, okay? Burns and warts. So you see the silver added to this Band-Aid. It's silver nitrate, and this Silvadine cream, 1% silver, or this silver compound here, silver sulfadiazanine. It's also used in wound dressing, especially for burns. Silver can have a good healing effect. It actually speeds wound healing for especially burns, but other wounds too. Copper sulfate can be used as an algicide. It can kill algae. So here, I think this was the Rio Olympics. Somebody got a stern talking to because they did not treat this Olympic swimming pool. They're doing a diving event here with copper sulfate. And look at this algae outbreak. Turn the water green. You only need this in low concentrations. We don't need a whole lot here. So, you know, it is an algicide that we use in the ecosystems and nature, and also here something like a pool. Okay. Next we have aldehydes. This is an aldehyde here, this aldehyde group over here. Aldehydes denature proteins and inactivate nucleic acids, DNA and RNA. They are bactericidal, virucidal, fungicidal, so killing outright. Long-term exposure, and it's over 10 hours or so, can even kill endospores, thereby achieving sterilization. How are they used? Again, pretty extensively in the medical industry. Cleaning medical equipment. This here, Sidex Plus. Look here, activated dialdehyde solution. Sterilizing and disinfecting. So cleaning medical equipment. It's used in embalming practices. Okay, aldehydes can be in, say, a morgue. Okay, or in the mortuary industry. Preservation of specimens like formaldehyde, you've probably heard of, to preserve specimens. There's aldehyde right there, formaldehyde. And then the last big one here is gaseous agents. And there's different gases that can be used, but the real big one I want you to know is ethylene oxide. Ethylene oxide denatures proteins and damages DNA, okay? After 4 to 18 hours, it can actually kill endospores, all right? It's one of the most common sterilization methods in the healthcare industry. We see a worker here. Why? Because ethylene oxide is used on instruments that cannot tolerate heat, moisture, or chemicals. Now, most everything we've mentioned so far has involved heat, moisture, or chemicals. So lots of things like these things, electronics, optical equipment, paper, rubber, cloth, plastics, bedding, things like that, can't tolerate high heat, moisture, or abrasive chemicals. They get destroyed. But ethylene oxide is quite effective to sterilize them. One of the most common sterilization methods in the healthcare industry, and the crew of the Apollo 11 back in the day used ethylene oxide to sterilize their equipment here. Finally, what about prions? We've talked about bacteria, fungi, viruses. Prions are by far the most difficult microbe to destroy. Endospores are second, but we've seen some things that can destroy endospores. Prions, remember, you might recall, are infectious proteins, just a protein that causes a refolding, a disrupting of normal proteins, especially brain proteins. Prions are very stable proteins, very difficult to destroy. They are inactivated by heating at 132 degrees Celsius or more for an hour. You might remember ultra pasteurization, you would get up to 132 degrees Celsius, but only for 1 to 2 seconds. You got to leave it that hot for an hour. And you need 30% bleach to destroy them. 10% kills bacteria and most other microbes. And they're also more resistant to radiation than living organisms. So prions can be very, very difficult to destroy. Okay? All right, everyone. So that is it for chemical control of microorganisms. Next time, we'll be looking at control of microorganisms in the body, inside living things. So the last two chapters, physical and chemical control, were in the environment. All right, everyone. So until next time, that's it for today. Have a nice rest of the day. Bye-bye.